Good morning from Texas. As Ala mentioned, my name is Martinez-Sobrido. I work at Texas Biomedical Research Institute, and I want to thank you for the invitations and the opportunity to present some of the research that we have been doing on SARS-CoV-2. And actually, I'm going to focus on the research that we have done during the last year, since we started at Texas Biomed. As Ella mentioned, we relocate our lab from University of Rochester to Texas Biomed exactly 1 year ago during the pandemic. Probably not the best time to relocate the lab and your home during the course of such a pandemic, but this is how things go. And research in our laboratory mainly focus on basic science, such as study virus-host interactions and mechanisms of viral pathogenesis. But we are trying to give our basic research a translational component, meaning we're trying to implement or use these findings in basic research or new approaches for the treatment of viral infections. And to answer some of these questions of basic research, or to develop these new approaches for the treatment of viral infections, you need to develop tools. And today I'm going to talk about one of these tools that we make in the lab to study SARS-CoV-2. That is the ability to generate recombinant viruses using reverse genetic approaches, and how we use these reverse genetic approaches to generate recombinant virus that express reporter genes that allow us to easily track the virus in vitro and also in vivo. And here I just show you some of the images that I will show you later of, in this case, mice infected with one of these recombinant SARS coronavirus that express luciferase, and how you can use this in vivo imaging system to track viral infection in the entire mouse at different days post-infection. And here on the right, some of the images of these fluorescent viruses that we make that allow you to see viral infection in culture cells as well as in vivo in the lungs of infected mice using these in vivo reverse genetics. So now, as Ella mentioned, before starting at Texas Biomed, or let's say before the pandemic, research on our lab mainly focused on influenza, rhinovirus, and Zika. The reason why we study influenza is because, besides it's significant in humans, is because at Rochester we have one of the centers of excellence on influenza research and surveillance. And our laboratory was responsible for the pandemic influenza response. The reason why we study arenaviruses is because several members of this family cause hemorrhagic fever in humans, mainly Lassa virus, and today we do not have yet vaccines or specific antivirals for the treatment of arenavirus infection. We started working with Zika virus back in 2015. Because, as you know, has been responsible of an outbreak of severe birth defects such as microcephaly and neurodegenerative disorders in babies that were born to infected mothers in North and South America, as well as in the Caribbean. But right after we moved to Texas, a new coronavirus that was called SARS-CoV-2 emerged in China, and since then he's been spreading around the world, and it's been responsible of this COVID-19 pandemic that is the worst pandemics that we have faced since the Spanish flu of 1918. So I'm sure that you guys are very familiar with SARS-CoV-2 and doesn't need that much introduction. I saw that several of you have been talking about SARS-CoV-2 and COVID-19. So the virus originally, as I mentioned before, originated in China. It's been spreading, and as to date has been on, and this is probably already updated, has been responsible of more than 145 million infections and over 3 million deaths around the world. Early this year, vaccines have been available, and these vaccines have been used in massive vaccine campaigns, and I hope you all have the opportunity to get one of these vaccines that have been approved for human use. But as you know, there is still an urgent need to develop more effective intervention, both prophylactic and therapeutics, for the treatment of SARS coronavirus. And this is particularly important right now because, as you know, there are the emerge of these new variants, viral variants called variants of interest or variants of concern. And it's still unclear if the current vaccines will protect against these new emerging variants of concern. And actually some research already suggests that some of the vaccines might not protect that efficiently against these newly emerged variants of concern. In this slide, I just want to show you a schematic representation of the. You see? Okay. Of the SARS coronavirus viral instruction here in panel A, as well as genome organization. So SARS-CoV-2 is a positive sense, single-stranded RNA viruses of approximately 30 kilobase. This is one of the largest RNA viruses that belongs to the coronavirus family. SARS-CoV-2 is very similar to other coronaviruses that are responsible of causing the common cold in humans, as well as these two previous coronaviruses that have been causing severe disease in humans, that is SARS-CoV-1 or SARS-CoV MERS coronavirus. Now, the viral RNA is transcribed in the cytoplasm of infected cells and they code for the major structural proteins that are shown here in panel A, the spike, the envelope, and the membrane protein, as well as different non-structural proteins from the open reading frame 1A and 1B, and other accessory open reading frame proteins shown here, open reading frame 3A, 6, 7A, 7B, 8, and 10 that are involved in viral replication, as well as in counteraction of innate immune responses during viral infection. The membrane of the virus is mainly made of the spike, envelope, and membrane protein. The spike, or S protein, is the receptor binding protein, is the protein that the virus used to enter the cells by binding to the cellular receptor that, as you probably are aware, is the angiotensin converting enzyme 2 protein. And the spike is also the major target of the neutralizing antibody. Actually, the goal behind the vaccines that are currently used in humans is to generate antibodies that can bind to the spike and neutralize viral infection. And here inside the viral of membrane, you have the viral RNA that is encapsulated with the nucleocapsid protein that, together with the RNA-dependent RNA polymerase encoded by the open reading frame 1A and 1B, is involved in viral replication and transcription. Now, we have been able to recently develop reverse genetic approaches to generate recombinant SARS coronavirus in the laboratory based on the use of bacterial artificial chromosome, or BAC. In this panel A here, you can see again a schematic representation of the viral genome with a unique restriction size that we use to assemble the entire viral genome as 5 segments or fragments, showing here fragment 1, 2, 3, 4, and 5 in this bacterial artificial chromosome. Now, in our case, since we did not have access to a natural isolate, what we did was to chemically synthesize the entire viral genome in these 5 fragments that we assembled together with this restriction size in the back. And when it's assembled, the viral RNA is expressed from the back from a polymerase II promoter. In our case, we use the cytomegalovirus or CMV polymerase II promoter, and is processed at the 3' end by the hepatitis delta virus ribozyme and the polyadenylation signal of the bovine growth hormone. And here you can see here how restriction analysis of this full assembled viral genome in the back with this unique restriction size. You can see the different fragments 2, 3, 4, and 5 that are assembled in the back. Now, in our case, we developed reverse genetic approaches to generate recombinant SARS based on this Washington 1 strain. And the reason why we use this Washington strain is because it was the first complete sequence available in PubMed. It was isolated from a fatal COVID-19 case here in the United States, in Seattle, Washington State. And because we knew that this will be the virus that will be distributed, at least for research purposes, from the CDC or BI resources at different laboratories. Now, once we assemble the entire viral genome in this bacterial artificial chromosome, we follow a very simple protocol to generate recombinant virus that consists on transfecting this unique plasmid directly into vivo E6 cells. And then 2 days later, we split the cells and put them in this T75 flask, and then evaluate the presence of the virus at 2 days post splitting the cells, by looking at cytopathic effect or by immunofluorescence using an antibody against the nucleocapsid protein. Now, we have been sharing these reverse genetic approaches already with over 80 laboratories around the world, and so far we have not heard any concerns about this. They all have been very successfully rescuing recombinant virus. So if somebody is interested in obtaining these reverse genetic techniques that we develop in the lab to generate recombinant virus, please send me an email and we will be very happy to share them. Now here, I just want to show you the results from the first rescue attempt that we did in the lab back in August 7, 2020. Where you can see here how the supernatant of transfected cells, they clearly show a cytopathic effect that it was comparable to cells that were infected with the natural isolate. And actually here also in panel B, you can see when we stain these infected cells with an antibody against the nucleocapsid protein, you can see how they are nicely staining and the level of infections are similar to when you infect them with the natural viral isolate. Now, one important thing when you develop reverse genetic approaches, either for SARS-CoV-2 or for other viruses, is to introduce a genetic tag or a genetic marker that allows you to distinguish the recombinant virus from the natural isolate. In our case, what we did was to introduce 2 silent mutations in the viral genome that remove 2 restriction sites. One is this restriction site, BST1, in the spike glycoprotein. The other one was a silent mutations in the membrane protein that remove MLE1 restriction site. So our recombinant virus will not have these restriction sites, contrary to the natural isolate that will contain these 2 restriction sites. So once we rescued the virus, what we did was to demonstrate that the recombinant nature of our virus, what we did was to amplify the membrane gene using RT-PCR approaches that I show you here from cells that were mock infected or infected with the recombinant virus and the natural isolate. And then this PCR product we digested with the MLE1 restriction site. And as we expected, the amplified gene from the natural isolate was digested with this restriction enzyme because it contains the IAM for this restriction protein. However, in the case of our recombinant virus, since we introduced this mutation that is shown here by Sanger sequence and removed the MLE1 site, we did not digest this product when we use MLE1 site. Now, these RT-PCR and Sanger sequencing results were further confirmed when we complete sequence the entire virus that we rescue using these reverse genetic approaches. And as you can see here, the only 2 mutations that we observe in the recombinant virus that we generate with this back-based system were the 2 restriction enzymes that we introduced to remove BST1 in the spike protein and MLE1 in the M protein that were actually present in the bacterial artificial chromosome that we use to generate the recombinant virus. Now, once we confirmed the recombinant nature, what we did was an in vitro characterization of our recombinant virus, shown here in red, and compare it to the natural isolate, shown here in black. To do that, we evaluate viral growth kinetics in vivo E6 cells infected at low multiplicity of infection at different time points, as well as plaque assay, as you show you here in panel B. And we observed is that the recombinant virus that we generate seems to have similar or identical variant fitness to the natural isolate, as you can see here by this growth kinetics, as well as plaque phenotype, demonstrating that we can rescue recombinant viruses using this back reverse genetic system, and at least in culture cells, our recombinant virus seems to behave very similarly to the natural isolate in viral growth kinetics and in plaque phenotype. Next, we evaluate our recombinant virus into rodent models that we developed here in Texas Biomed, and they have been used also for many other laboratories around the world. One of them is the golden Syrian hamster model, and the other one is the K18 human AC2 transgenic mouse model. Now, here in the case of the hamster model, what we did was to infect them with 10 to the 5 PFUs of either the recombinant virus that we generate by reverse genetics or the natural isolate, and then evaluate viral replication in the lungs in panel B, or in the nasal turbinates in panel C at days 2 and 4 post-infection. And as you can see here, our recombinant virus seems to replicate very similar to the natural isolate, either in the lungs or in nasal turbinates at days 2 and 4 post-infection. And these results in the hamster model were further confirmed with the K18 human AC2 transgenic mouse model. As you probably know, in this mouse model, animals that get infected with SARS-CoV-2 rapidly lose weight and succumb to viral infections between days 6 and 8 post-infection, with high viral titers in the nasal turbinate and in the lungs. And as you can see here, when we infect this transgenic mouse model with our recombinant virus, showing here with these triangles, or the natural isolate, animals rapidly lose weight, and all of them die between days 6 and 7 post-infection, with similar replication in the nasal turbinate and in the lungs at days 2 or 4 post-infection than the natural viral isolate. So to summarize this first part of the presentation, we have been able to clone the entire genome of the SARS coronavirus 2 Washington strain in the backbone of a bacterial artificial that we have been used to generate recombinant viruses. We have confirmed the recombinant nature of our recombinant virus, and what is important is that the virus that we generate using this reverse genetics seems to have a very similar fitness than the natural isolate in vitro, by looking at plaque phenotype and growth kinetics, and in vivo, both in the hamster model and in the transgenic mouse models of infection, where the virus, recombinant virus, has similar pathogenicity and replicate to the same extent than the natural isolate. And you might be wondering why we're so interested in developing these reverse genetic techniques to generate recombinant virus. And the reason is because reverse genetic techniques are probably, if not the most powerful tool in modern virology, since allow you to generate recombinant viruses in the lab, not only wild type, but as I saw you in the previous part of the presentation, but also recombinant virus containing mutations, and allow you to answer important questions in the biology of SARS-CoV-2. For instance, we can now use these reverse genetic approaches to study the functions of viral proteins and their contribution in viral pathogenesis. Actually, we have been able to generate already recombinant viruses where we delete individually every single open reading frame, like the open reading frame 3A, 6, 7A, 7B, and 8, and assess the contribution of these accessory proteins in pathogenesis using the transgenic mouse model of infection. You can also use them to introduce amino acid mutations in a particular viral protein to assess their contribution in replication and pathogenesis, to understand the role of untranslated regions in viral replication, to generate, similarly to the influenza virus, attenuated forms that they can use as live attenuated vaccines, or to generate recombinant viruses that express reporter genes to facilitate the study of SARS-CoV-2 in vitro and also in vivo. And we are actually using these reverse genetic approaches to do all these things. But today, for my presentation, I'm going to focus on the use of these reverse genetic approaches to generate recombinant viruses expressing reporter genes. So as you probably know, one of the limitations to study SARS-CoV-2 as well as many other viruses is the need of secondary approaches that allow you to identify infected cells, such as antibodies. However, we and others have shown how recombinant viruses expressing reporter genes represent an excellent option to track viral infection in culture cells as well as in animal models without the need of these secondary approaches like antibodies to identify what cells are infected. So one of the first things that we did when we developed these reverse genetic approaches was to generate recombinant viruses expressing fluorescent and luciferase proteins. In the case of fluorescent proteins, we use Venus, that is a monomeric form of the yellow fluorescent protein, as well as the monomeric cherry protein. And in case of luciferase, we make recombinant viruses expressing a nano-look or N-look luciferase. And to do that, we use an approach that has been shown previously to be working for SARS-CoV-2, where you substitute the reporter gene for the open reading frame 7A of the virus. And as you can see here, once we introduce the reporter gene in place of the open reading frame 7A of the virus and the bacterial artificial chromosome and transfect cells, you can see here we were able to efficiently recover viruses that express in this green Venus fluorescent protein or in cherry, as well as nanolook that I'm not going to show you now. And you can easily identify the presence of the virus in infected cells directly by using fluorescent microscopy without the need of antibodies to identify the presence of the virus in infected cells like you need for in the case of the wild-type virus. And in this case, actually, the fluorescent expression or the luciferase expression can be used as a valid surrogate to identify infected cells. And we have been using these viruses either with the fluorescent or the luciferase to evaluate neutralizing antibodies, as I will show you in a little bit, as well as antivirals against SARS-CoV-2. Importantly, when we evaluate the ability of these recombinant viruses expressing these reporter genes in growth kinetics and compare them to the wild-type virus, what we observe is that the expression of the reporter gene didn't seem to affect viral fitness because, as you can see here, viral cells infected a low MOI and assesses at different times post-infection with these reporter expressing virus seems to replicate very similarly to the wild-type virus that does not express the reporter gene. But one of the limitations of this first generation, we call it first generation, of reporter expressing SARS-CoV-2 is that, as you remember, we substitute the open reading frame 7A of the virus for the reporter gene. And although in this case doesn't seem to affect viral fitness in culture cells, as I just showed you in the previous slide, we didn't know if that will be the case in vivo. Another limitation of this approach is that the reporter gene is expressed from the open reading frame 7A. And we did not see that such a great levels of reporter gene expression. And for these two reasons, we decide to generate a new generation of reporter genes that hopefully express higher levels of reporter expression and that do not need to remove the open reading frame 7A from the virus. And to do that, we use an experimental approach that we previously used to generate recombinant influenza viruses expressing fluorescent proteins that is based on the use of the porcine testcerovirus 2A autoproteolytic cleavage site. And as you can see here, when we generate this recombinant influenza virus using this 2A approach, you can easily track viral infection in culture cells as well as in ex vivo by looking at fluorescent expression on the lungs of infected mice. And that's what we did. So in the case of SARS-CoV-2, we use again this 2A approach And what we did was to put the reporter gene, in this case shown here with Venus, in the locus of the nucleocapsid protein fused by this 2A autoproteolytic cleavage site. And the reason to select the nucleocapsid protein is because, similar to other RNA viruses, the nucleocapsid protein is probably one of the most abundant proteins produced during viral infection. And importantly, as I mentioned before, using this 2A approach, we do not need to remove the open reading frame 7A as we did before. And as you can see here in panel B, we were able to successfully rescue recombinant viruses expressing Venus from the locus of the nucleocapsid protein using this 2A approach. And one of the things that we observed right away is that it seems like the reporter Venus expression from the locus of the nucleocapsid was much higher than the initial virus that we generate from the locus of the open reading frame 7A. Now, to confirm that it was the case, what we did was infect viral cells with either the virus that expressed the Venus from the open reading frame 7A or the virus that expressed Venus from the nucleocapsid locus, and analyze the reporter gene expression Venus at different times post-infection by fluorescent microscopy here showing panel C, or by Western blot. And as you can see here, either by immunofluorescent microscopy or Western blot, you can see how the levels of Venus expression are much higher in the case of the virus that expressed it from the locus of the nucleocapsid protein than from the locus of the open reading frame 7A, both by fluorescent or Western blot. Importantly, when we look at the viral plaques from these two viruses using a chemidoc, what you can see here is how we can easily detect how the fluorescent expression of these viral plaques when the reporter gene is cloned in the locus of the nucleocapsid protein, and we could not detect it from the locus of the open reading frame 7A. And again, this is most likely because of the much higher levels of expression of the reporter gene in the locus of the nucleocapsid than from the open reading frame 7A. But what it was important is when we assess the replication or the kinetics of these reporter viruses and compare them to the wild type, we can see here that all of them replicate similar to the wild type, suggesting that expression of these reporter Venus did not seem to affect viral fitness, at least in vitro, in viroid cells. In vitro studies, we were very encouraging to evaluate if that was the case also when we infect the transgenic K18 mice. So in this case, what we did was to infect the K18 transgenic AC2 mice with the wild type virus or the two viruses that express Venus, either from the open reading frame 7A or from the NP locus, and assess for a fluorescent expression in the lungs of infected mice at days 1, 2, 4, and 6 post-infection. And as you can see here, we can easily detect Venus expression from the lungs of infected mice with the Venus that express in the locus of the nucleocapsid protein at all days post-infection. Unfortunately, that was not the case of the mice that were infected with the virus expressing Venus from the open reading frame 7A, further suggesting that the levels of expression from the nucleocapsid locus are much higher than those of the open reading frame 7A. What it was very important is when we evaluate the replication of this virus and compare them to the wild type, either in the nasal turbinate, in the lungs, or in the brains of infected mice at days 1, 2, 4, and 6 post-infection. The virus expressing the Venus, either from the open reading frame 7A or from the NP locus, replicate very similar or identically to those of the wild type. Importantly, all the mice that were infected with this recombinant virus expressing Venus, all of them rapidly lose weight and succumb to viral infections between day 7 and 9, similar to the virus, recombinant virus that wild type that do not express, suggesting that the expression of these reporter genes from either the locus of the open reading frame 7A or NP do not affect pathogenesis of the virus in this animal model. Now, these results were very encouraging. Say we need to make now a virus that expresses nanologue, and that's what we did. We used the same two-way approach and expressed nanologue from the locus of MP. And one of the first things that we did, similar to the Venus, is that the recombinant virus expressing nanologue from the nucleocapsid locus expressed like 50 times more than the virus expressing the nanologue from the open reading frame 7A, and expression of this nanologue did not affect fitness in cultured viral cells, since the virus were able to replicate very similarly to the recombinant and wild-type virus. And next, we test the feasibility of using this nanologue recombinant virus to evaluate the viral infection in vivo using an in vivo imaging system. And that's what I show you here. So to do that, what we did was to infect against the transgenic human AC2 mice with either the recombinant virus that express nanologue from the MP locus or the recombinant wild-type virus, and assess for luciferase expression and different times post-infection. And as you can see here, we were able to nicely detect the expression of nanologue, in this case, using the entire mouse at all the time days post-infection, as I show you here, with the entire mouse, except for day 6, because one of the mice here already died from infection with this reporter virus expressing nanologue. What it was important is, and similar to what it happened to the Venus virus, expression of nanologue did not affect the pathogenicity of the recombinant virus. As you can see here, all the mice infected with the recombinant virus expressing nanologue rapidly lose weight, similar to mice infected with the wild-type virus, and all of them succumb to viral infections between day 6 to 8. And what it was important is that the replication was in the lungs, nasal turbinate, and brains was also very similar to the wild-type virus. And importantly, we were able to detect nanoluciferase expression in these tissues, in the nasal turbinate and lungs and brain, of the mice infected with the nanologue, and obviously we did not detect it in the case of mice that were infected with the recombinant wild-type virus. Now the question is, can we use this nanoluciferase expressing virus to evaluate therapeutics against SARS-CoV-2? And the answer is yes. And to demonstrate that, what we did was to use a monoclonal antibody that we identified and isolate from a human that was infected with SARS-CoV-2 that is called 1212C2, and we showed that nicely neutralized the virus in vitro. So what we did was, again, using the transgenic mouse model, we treat mice with either this neutralizing antibody, 1212C2, or the isotype control, IgG, and then infecting with the reporter expressing nanologue virus, and assess for a viral replication using this in vivo imaging system at days 1, 2, 4, and 6 post-infection. And as you can see here, mice that were treated with this 1212C2 monoclonal antibody, we could not detect luciferase expression, suggesting that this antibody clearly neutralized the virus, not only in vitro, but also in vivo. And that was not the case of mice that were treated with the isotype control and infected with luciferase, because as you can see here, all the mice were expressing luciferase that obviously increased in a time-dependent manner. So suggested that we can now use this reporter, in this case luciferase expressing virus, and this in vivo imaging system to directly evaluate in vivo the ability of antibodies or antivirals to neutralize or to inhibit viral infection. So as you probably are aware, and I mentioned earlier, one of the major concerns with the current COVID-19 pandemic is the emerging of these viral variants called variants of interest or variants of concern that have been identified already in different parts of the world, including UK, South Africa, Brazil, United States, and India. And the reason is, again, because we don't know if the vaccines that we are currently using are going to be able to protect or protect as efficiently against these variants of concern as they do with the original Wuhan virus. So what we did now, and this is the work from a graduate student, and I think it's very interesting. What we did now is to generate a recombinant virus that now express instead of Venus and cherry, and contain the 3 mutations in the spike receptor binding domain, or RBD, that were identified in this South African variant of concern, that are mutations at position 417, 484, and 501. And here in panel B, I just show you the Sanger sequencing showing the mutations at these different positions. So now we have 2 viruses. We have the original virus that express Venus. Now we have this new virus that express cherry that contains the mutations of this South African variant of concern. And what we thought is that it would be nice to implement a bifluorescent microneutralization assay that allow us to identify at the same time if, for instance, a monoclonal antibody or a sera from an infected patient or a vaccinated patient is able to neutralize the original virus or this new variant of concern. And that's what I tried to show you here. So what we did is to either incubate the original virus, Washington strain that is present Venus, with this variant of concern that is present cherry, with monoclonal antibodies, and then infect cells, with the understanding that if the antibodies do not neutralize the virus will be able to infect the cells, and we will see fluorescent Venus and cherry expression. However, if the monoclonal antibody or the human sera is able to neutralize both viruses, the virus will not be able to infect the cells, and we will not see expression of either Venus or cherry. However, if the virus is able to neutralize specifically one virus, but not the other, we will see expression of one fluorescent protein, in this case red, if it does not neutralize the South African variant of concern, of green, in the case that does neutralize the original Washington strain virus. And here I want to show you the results of this bifluorescent microneutralization assay using infection with this virus individually or in combination. And to do that, we use 2 monoclonal antibodies, the 1212C2 that I showed you before is able to efficiently neutralize original Washington strain, but we found out that it's not able to neutralize the South African variant of concern. And another antibody that we use is this 121387, that is a monoclonal antibody that we also isolate in the lab from a patient that was, sorry, I don't know what happened. Technology problems, I guess. What I was saying is that we developed this bifluorescent microneutralization assay that I will show you later. We try to span a multiplex fluorescent assay to identify neutralizing antibodies that are able to specifically neutralize either the original virus expressing one fluorescent protein versus one of these newly identified variant of concern. In this case, we use the South African variant that expresses a different color, in this case, cherry. And as proof of concept, we use these 2 monoclonal antibodies, one that we know that specifically neutralizes the original strain, but not the variant of concern, and this 121387 that we know that neutralize both the original virus and the variant of concerns. And as you can see here, this 1212C2 nicely neutralized in a dose-dependent manner the original virus, but not the South African variant, either when you infect individually cells with these 2 viruses, or when you infect at the same time the same cells with the 2 viruses at the same time. You can see how 1212C2 nicely neutralized the original Washington strain, showing here in green, but not the South African variant. While this 121387 monoclonal antibody that is able to neutralize both, you can see here the original Washington as well as the variant of concern, South Africa, in cells individually infected, with these two viruses separately, or when you co-infect cells with the Venus Washington and the M. cherry South Africa. So that was great, but the question is, can we do this in vivo? I mean, can we infect mice with these two viruses and treat them with antibodies and see if one of them specifically neutralize one strain and not the other, or neutralize both of them, or none of them? By looking at the lungs of the infected mice with these two viruses using this in vivo imaging system. And this is what we did. I just want to show you here the results that we obtained in the case of mice that were infected with the Washington strain virus that express Venus. As we expect, the mice that were treated with either 12-1387 or 12-12C2, that we know they both neutralize the original Washington strains, we were not able to detect fluorescent Venus expression either at days 2 or at day 4 in the lungs of these infected mice, contrary to the situation of mice that were infected and treated with isotype control that does not neutralize the virus. And that's what you see Venus expression at days 2 and day 4. In the case of mice that were infected with the South African variant that is present cherry, and similar to what we see in our in vitro studies, 12-1387 is able to bind the virus and neutralize, so that's why we do not see M-cherry expression at either day 2 or day 4 in the lungs of infected mice. In the case of 12-12C2 monoclonal antibodies, since we know that does neutralize this virus, we can see M-cherry expression in the lungs of infected mice, and the levels of expression were very similar to those that we observed in mice that were infected or treated with the isotype control. Now, what it was very interested is when we evaluate the lungs of the mice that were infected at the same time with the Washington original strain expressing Venus and the South African expressing M. cherry. As you can see here, the mice that were treated with 12-1387, since this monoclonal antibody is able to neutralize both of the virus, we could not detect Venus or M. cherry expression either at day 2 or at day 4 was challenged with the virus. In the case of mice treated with 12-12C2, that is only able to neutralize the original virus, but not the South African isolate, we could detect expression of M. cherry, but not M. Venus at days 2 and 4 post-infection. And as we expected, in the case of mice that were treated with the isotype control and infected with the two viruses at the same time, we were able to detect both the original Washington virus expressing Venus, as well as the South African variant expressing M. cherry. And importantly, when we evaluate the body weight and showing here in panel A, as well as the percentage of survival in these mice, that we found is that, as we expected, the mice that were infected with the Washington Venus expressing virus that were treated with both either the 12-12C2 or the 12-1387, they all maintain their body weight, and they survive infection because these two antibodies are able to neutralize the Washington strain. However, that was not the case in mice treated with the isotype control that rapidly lose weight and die between day 6 and 8 post-infection. In the case of mice that were infected with the South African variant, as we saw in our in vivo imaging system, only the mice that were treated with 12-1387 maintained their body weight and survived infection, contrary to mice that were treated with 12-12C2 that rapidly lose weight and die from infection, similar to mice treated with the isotype control antibody, and it's because this 12-12C2 is not able to neutralize the South African variant. And as we expect, in mice that were co-infected with the two viruses viruses at the same time, the Washington expressing Venus and the South African expressing M. cherry. Only those treated with the 12-13-87 monoclonal antibody survive infection, while those treated with the 12-12C2, they lose weight and succumb, similar to the mice treated with the isotype control. Now, based on these results that we have so far with these two viruses, that is the Washington strain expressing Venus or the South African variant of concern expressing in cherry, what we are currently doing now is generating other recombinant viruses expressing the spike of other variants of concerns, mainly focused now in this Delta Indian variant that now express a different fluorescent protein, in this case, a cyan fluorescent protein, and develop a multifluorescent microneutralization assay that hopefully will allow us to evaluate not only in vitro, in culture cells, but also in the K18 mouse model, as I showed you before, the ability of human sera or monoclonal antibodies or drug therapies to neutralize or to inhibit infection, not only for the Washington strain or original strain of SARS-CoV-2, but also against this new emerging variant of concerns. And I just wanna finish here thanking the people in the lab that they actually deserve all the credit because they are the ones that did all the work and they are the real COVID-19 warriors. Especially I want to thank Chen Jing, that is the mastermind behind all these reverse genetics and the generation of all these recombinant viruses, as many as others that we are generating and using in the lab, including attenuated forms of the virus. June, that is the major driving force of the in vivo study, and the two graduate students, Kevin and Desiree, that have been responsible of characterizing all these fluorescent recombinant viruses. And I'm going to leave it here. Thank you again for the invitation. Sorry for technology challenge, and I will be happy to answer any questions.
Thank you so much for the talk, and we are happy that you could finish the talk.
Thank you.
And there is time for a question. Pavel?
Hi, Luis. Sorry for finishing up too early, and I'm really happy that you're able to join us. I'm wondering, did you test the stability of your reporter viruses through the passages? Maybe it's not very relevant for the in vitro cell culture, but it will be really relevant for your in vivo models, since it could change, and maybe your inhibition or no signals, it's not really due to the inhibitors or antibody, but it's because of the virus is lacking the reporter.
That's a great question. That's an excellent question, and I'm glad you asked it. Yes, we evaluate the stability of these viruses, not only in passaging in vitro, multiple passage, but also the virus that we recovered from these infected mice at different days, 1, 2, 4, and 6. Now, what we found out is that the virus that expressed the reporter gene from the locus of the nucleocapsid protein, since the nucleocapsid protein is completely necessary for the virus for encapsulate the viral RNA and for replication, we found these viruses that express the reporter gene from the nucleocapsid locus being very highly stable, not much more than those that express the reporter gene from the open reading frame 7A, because that's an accessory protein that actually we know the virus would not need, not because of the in vitro studies that we make in the lab with the recombinant virus, but also natural isolates that has been identified already in humans that do contain deletions or mutations or truncations of this open reading frame 7A. That's another advantage of using this two-way approach and expressing the reporter genes from the nucleocapsid. Not only that you do not delete any viral protein, or you increase also the levels of expression, also that you increase the stability of these reporter viruses.
I got also one more question.
Sure.
Are there any other factors which affect the effective expression of the reporter gene? I mean, the sequence and structure of the construct.
That's a great question. Again, so far we have only made viruses that express these two fluorescent, the monomeric form of the yellow fluorescent protein, the Venus, or the M-cherry. And the reason why we use these two fluorescent proteins is because of previous studies that we did with influenza, where we use the same reporters. But we have not evaluated yet if different reporters will have or not different impact on the virus.
Okay. And one question from the chat. Have you already tried to use your system to test any small molecules, drugs, that could potentially target SARS-CoV-2?
Yeah, that's a great question. I mean, since we developed these reporter viruses, and as you can imagine, they are more compatible with high throughput screening approaches. We are testing antivirals, we are testing neutralizing antibodies, with the advantage of you can use this reporter as a valid surrogate of viral infection, meaning you don't need to use like an antibody to detect the presence of the virus. So yes, as I show you, we are using now pretty much all the time these reporter viruses, either or using fluorescent or luciferase to evaluate antivirals and neutralizing antibodies. And the idea will be to use them not only for the original Washington or Wuhan strain, but also with these new variants of concern that are emerging.
Thank you. Any more questions, comments? I don't see. So thank you again. It was nice to host you online, but hopefully next time we will be here in Poznań finally. Thank you.
Okay. Thank you so much.